So good morning, everyone. Welcome to the BEEc Speaker Series. This is going to be our first talk of the season, so we're very excited to have this talk today. I'm just going to give a little quick spiel, just some logistics. Just a little background first about BEC. So BEC is the Center for Bee Ecology, Evolution, and Conservation at York University. We are an organized research unit, and if you'd like to learn more about us, you can visit our website at www.yorku.ca/bees, or you can email me, the BC coordinator, at bc@yorku.ca. So we are an initiative that strives to advance research in the fields of bee ecology, evolution, and conservation. BC's mission is to foster interdisciplinary, innovative, collaborative, and cutting-edge research to advance our knowledge on bees and implement policy changes to help sustain pollinators globally. Our goal is to apply our collaborative efforts to the development of policies and environmental management for the long-term sustainability of bees and the vital ecosystem services they provide. And if you'd like to get our monthly newsletter, you can also subscribe to our listserv. You can send an email to listserv@yorku.ca, and with the following comment in the body of the email, you can write subscribe bees along with your first name and your last name. This will also allow you to get our monthly newsletter and information on any other upcoming events. Also, if you are a York University researcher or collaborator, this includes graduate students, please visit our website to see if you are eligible to apply to be an associate of BC. And the pictures down below here are just some of the examples of some of our B associates. And so we have some new upcoming talks for our series. So keeping in mind, we do not have a seminar series for October, as we will be hosting our annual conference instead, BEEcon. Just as a reminder, registration is still open for that if you are interested. But the BEEc Speaker Series will typically take place once a month, usually the second or third Friday of that month, and it's always at 11 a.m. Eastern. You can register for this series on our website, and the registration for the next talk in November is also currently open as well. And we're in the process of finding another speaker for December. And this will also continue in January to April. So just a couple more slides on logistics. So for today, we are using the Zoom platform, and only panelists can turn their camera and microphones on. We ask that you please use the Q&A box to pose any questions that you may have for the speaker, and please to do so at the end of the talk, and we will pose those questions to the speaker towards the end. Do use the chat box, although I don't think it's working today, sparingly if you have any general comments, as it can be distracting. But again, if you do have any questions, try to just use the Q&A box, but only towards the end after the talk is done. If you're experiencing any issues, you can send a private message to myself, or you can email bc@yorku.ca. Also, if you do happen to need to leave at the hour mark, so this will end at 12 when we're still in the Q&A period, don't worry, because this presentation and the Q&A period will be recorded and posted on YouTube at a later time. And so you will have a chance to at least look at the talk later and any questions that may have been asked. The talks are also going to be posted on YouTube, as I mentioned, so you can visit youtube.com/@beesyork. We have several previous talks already available on the YouTube channel to view. We also have talks posted from other programs like the BEEc Associate Series, as well as BEEcon, our annual conference, and you can also subscribe to our channel and be notified if there's any new videos posted. And before we begin the talk, I would like to recognize that many Indigenous nations have long-standing relationships with the territories upon which York University campuses are located that precede the establishment of York University. York University acknowledges its presence on the traditional territory of many Indigenous nations. The area known as Takaranto has been caretaken by the Anishinaabe Nation, the Haudenosaunee Confederacy, and the Huron-Wendat and the Métis. It is now home to many Indigenous peoples, and we acknowledge the current treaty holders, the Mississaugas of the Credit First Nation. This territory is subject of the Dish with One Spoon, Wampum Belt Covenant, an agreement to peaceably share and care for the Great Lakes region. If you'd like to learn more about the native land in the area that you work in, or just for more information, you can always visit www.native-land.ca to expand on the background of the area that you're working in. And with that, today I'd like to welcome our first speaker of the series, Dr. Clara Stuligross. She is an assistant professor of biology at Kalamazoo College in Michigan. She earned her PhD in ecology at the University of California, Davis, working with Neil Williams, and was a postdoctoral fellow at the USDA National Institute of Food and Agriculture at UC Riverside with Hollis Woodard and Nicole Rafferty. Clara's research focuses on the impacts of environmental stressors on wild bee ecology, with particular emphasis on pesticide exposure, floral resource availability, and climate warming. She's also interested in developing conservation strategies to help mitigate these stressors in both natural and managed landscapes. And so today, Clara will be talking about the impacts of environmental stressors across life stages of wild bees. Go ahead.
Thank you so much for the introduction and also for inviting me to speak to you all today. So today I'm going to give a brief introduction to some major environmental stressors that affect bees. And then I'm going to talk about some of my past research on the impacts of pesticides and floral resource availability on bees. And then I will talk about some of my more recent work on climate change and heat waves and their impacts on bees. All right. Perfect. So, human actions have fundamentally altered the environment, and today much of our landscapes have been transformed. And this is happening all across the world, but in particular, agricultural landscapes have seen some of the most dramatic changes over the past several centuries. And these changes are predicted to accelerate. And as you can imagine, this really affects the organisms that live in these environments. And we are particularly interested in how bees are affected by these environmental changes because of their role in pollination. And as all of you, I'm sure, know, pollination is really vitally important to terrestrial ecosystems and to crop production. Almost 90% of flowering plant species rely on animal pollination. And from a more human-centric perspective, over 70% of global crop species also rely on pollination, and pollination services in general are valued at about $215 billion each year globally. And much of this pollination is done by both wild and managed bee species. And in many cases, there's sort of this irony where these intensive areas of intensive agriculture that really depend on pollination services from bees also present considerable challenges to bees that are living in these areas. So bees experience multiple stressors within agricultural landscapes, such as habitat loss, degradation, and fragmentation, as well as an interacting combination of pesticides, parasites, and disease. And they can be exposed to these risks simultaneously as they forage widely for pollen and nectar resources. In my research, I'm really interested in understanding how these stressors affect bees. And so far, I've primarily focused on these three stressors. So the impacts of food abundance, pesticide exposure, and climate change on bees. And so right now, I'm going to spend a little bit of time talking about the work that I've done on food and pesticide stressors. And then later on in the talk, I will talk more about climate change. So bees, like all organisms, need food, and for bees, this comes in the form of pollen and nectar from flowers. So habitat loss has led to decreased floral resources across landscapes and subsequent bee declines. And without these abundant and diverse flower resources, food stress can directly reduce offspring production in bees. It can also impact just overall survival, but it can also increase bees' susceptibility to other types of stressors. And when we think about pesticides, bees can be exposed to these pesticides directly during application. They can also contact residues of pesticides on plants or nest materials, or by consuming contaminated pollen and nectar. And this type of exposure can cause direct mortality. It also can have sublethal effects like reduced reproduction or slower development or impaired foraging behavior and learning. We also know very little about the interactions between stressors and what might happen when bees experience these stressors simultaneously. So, for example, bees that are exposed to pesticides might be less efficient at foraging, and that might make them even less able to collect food resources. Or if they have abundant food resources, that could offset the negative effects of pesticide exposure. But in addition to not knowing that much about the interactions between stressors, we actually don't know very much about the impacts of stressors in general on the vast majority of bees. And that's because most research focuses on the impacts of individual stressors on just a single bee species, primarily the Western honeybee, despite the fact that there are over 20,000 bee species worldwide, including 465 bee species here in Michigan, where I work, and over 800 species in Canada. So there's a really huge diversity of bees, and we don't know that much about almost all of them. So I'm sure many of you in the audience are very familiar with solitary bees, but for those of you who don't know, over 80% of bee species are solitary, which means they don't live in hives or colonies like social bees, but rather each individual female bee is solely responsible for collecting resources and providing for her offspring. There is a really big diversity in different types of nesting styles. So some bees nest in pre-existing cavities like holes in wood, as you can see here in the photo. Some will excavate their nests out of the soil, and some even will use other materials like flower petals or leaves to encase their eggs for protection. And so we really know very little about how solitary bees respond to stress, even though they comprise the vast majority of bee species. And so in my research, I'm trying to work to fill some of these gaps in our understanding about the impacts of multiple stressors on bees with a focus on solitary bees. So I'm going to share with you today some stories about some of the research that I have done. First, to explore the effects of pesticide exposure and resource limitation on solitary bee reproduction, and also on the impacts of carryover effects of past pesticide exposure on solitary bees. So for these studies, I looked at what is, I think, becoming a model for solitary bee biology, which is the blue orchard bee, Osmia lignaria. These bees are solitary. They're found pretty widely throughout the United States, including here in Michigan, also near you all at York University. And they exist in wild populations, but they're also used specifically for agriculture, where they are released in large numbers, especially in places like fruit orchards or nut orchards like cherries and almonds, where they are used to increase crop pollination in those areas. So I'm going to spend a few minutes going over their general biology and especially their life cycle, which will help to understand the type of research that I do. So the life cycle of Osmia starts in early spring. So adult bees will emerge from their nests where they spent the winter in diapause, which is sort of like a hibernation state. Male and female bees will emerge and mate. And then the female bee will start collecting pollen and nectar from flowers. These bees find pre-existing tunnels for their nests, such as things like old beetle burrows in trees or hollowed out stems, and they use those cavities to create their nests. And this is really convenient for research because we can use these wooden nesting blocks with holes drilled in them to provide this tunnel shape that they really like to use. You may have seen these places, they're sometimes called bee hotels, and they kind of mimic that natural cavity shape that a lot of these cavity nesting bees use. And so over the next several weeks, the mother bee will collect this pollen and nectar and put it inside of her nest. You can see here on this image on the top, this is a cross section of an osmia nest where you can see those purple pollen balls are those sort of pollen and nectar provisions that the mother bee collected. And then on top of each one of those, she will lay an egg and then seal each of those cells with mud to protect the baby bees inside. And the use of mud in their nests is what gives these bees the nickname mason bees. And you can see here in this little video, this bee is sealing up the end of her nest with mud, which is really important for the protection of the nest. And so these eggs over the next couple of weeks will hatch into larvae. They will eat the food that their mother left for them and continue to grow over the next few weeks. So you can see here that in this video, this is a bee larva eating that pollen provision. The flowers that I use in this research have purple pollen, so all of these have this really bright purple pollen, which I think is cool. And then after they finish their provision, these bees will spin a cocoon around themselves and pupate. And then throughout the fall and winter, the bees will develop into adults and remain sealed inside their nests within their cocoons until springtime. And then when it starts to warm up, the new bees will emerge and start the cycle all over again. So this whole life cycle takes about a year. And one really great aspect about these bees is that we can actually study each of these life stages both in the field and also in the lab, so we can really get a sense of what is happening to them throughout their lives. So now I'm going to go into some methods that I used in this first study to look at the effects of pesticide exposure and resource limitation on solitary bee reproduction. So I designed a field experiment using these field cages. So this allowed me to expose these bees to stressors in a controlled way while still maintaining some of the natural things about being outside. So you can see in these photos, this is a photo of all the cages that I used. This research was done in California. And so cages received a crossed resource by pesticide design. So first, cages received, they had flowers at either high densities or low densities. So you can see here in the photos on the left show cages with essentially unlimited floral resources. And then on the right, you can see pretty limited floral resources. So they either had a lot of resources or limited resources in those two treatments. Cages were also treated with or without pesticides. So half of the cages had this pesticide stress and half of the cages had this resource stress, and we had each combination of those treatments. And so in each of these cages, I placed a nesting block and released bees, and I had a total of 4 cages per treatment. Because all the bees in these cages look basically the same to my untrained human eye, I used little paint marks so I could connect each individual female with her offspring and kind of tell them apart and see what they were doing individually. And this allowed me to measure things like demographic effects over the season, counting the reproduction and nesting activity of each individual bee. So I'm going to show you a series of results that all look something like this. So the food resource availability treatment is indicated on the x-axis with low resources on the left, high resources on the right. And for the first graph, we are going to look at the number of surviving offspring per nesting female on that y-axis. And so first we'll look at the values for the control cages, which did not have any pesticide on them. So you can see this is basically the average number of offspring that each female had in those control cages in both of those resource treatments. And now we can see the data collected from the pesticide-treated bees, which is shown in red. So we found that both resource and pesticide stressors reduced reproductive success among these nesting females. So of those female bees that initiated nesting, those that were exposed to pesticides provisioned 42% fewer offspring than controls. So they had a 42% decline in reproduction. And bees with low resources provisioned 26% fewer offspring than those with abundant resources. So there was a 26% decline in reproduction from resource limitation. We also looked at the sex ratio of these offspring. So essentially the proportion of female offspring provisioned by each nesting bee. So both stressors led to what we consider to be male-biased sex ratios, which resulted in a significantly lower proportion of females overall compared to the control bees that had no stressors. So pesticide exposure caused a 33% reduction in the proportion of daughters produced, and resource limitation caused a 48% reduction in that proportion of daughters produced. And in this species, Osmiolignaria, the female bees are larger and they require more food. So those provisions that the mother provides for them are bigger. So they're essentially more expensive to produce. So what we're seeing here is that the females, when they're stressed by both resources and pesticides, they shift their reproduction to produce less costly male offspring. And this is another really clear sign of stress in these bees. So if we look at these results together, we see that both stressors pretty dramatically impact multiple vital rates in these bees by reducing their reproduction and reducing the overall number of female offspring. So I want to take a minute to put these results in perspective with the potential effects on population growth. So what does it actually mean that they're having fewer offspring and fewer female offspring? So if you remember from the life cycle, the females are what we consider to be the demographically important sex. So males don't contribute to the efforts of reproduction beyond that mating stage. So it's really important to consider both the effects on reproduction overall, but also the effects on sex ratio when we're thinking about population growth altogether. So if we consider the average female in an optimal environment with abundant pesticide-free forage resources, can produce, according to our data, about 37 offspring in her lifetime. And based on the sex ratios we observed in this study, about 10 of these offspring are female. So this is sort of the normal baseline condition in unstressed bees. On the other hand, bees that were double stressed, so they had pesticide stress and food stress, produced about 16 offspring each, which is a difference of 57% compared to that baseline. And when we look at the sex ratio, only 1 to 2 of those offspring are females. So this difference is really striking considering that even minor changes in offspring production can really substantially influence population growth, given the relatively low reproductive rate in solitary bees. And effects like this could reduce the reproductive potential of future generations, driving populations to declines or even to extinctions. So in summary, for this first study, we found that pesticide and resource stressors reduced reproduction and altered the offspring sex ratio, with some implications for future population growth in this type of bee. So now I want to talk about this second study that I did to investigate the carryover effects of past pesticide exposure on solitary bees. So what do I actually mean by that? I want to go back to our bee life cycle. So, as I mentioned earlier, most studies look at the effects of stress on just one life stage. In the study I just described, that's exactly what I did. We looked at the effects of pesticide exposure to adult bees and looked at how it affected their reproduction. So, just that one life stage. This is really important work, but if we think about a real world context, bees are exposed to stressors at multiple stages of the life cycle. So we can't expect that just looking at reproduction from adult exposure alone will tell us the full story. So for example, when this mother bee forages on a flower that has been contaminated with pesticides, the bee herself becomes exposed, and this could influence her capacity to nest and to provision offspring, which is what we saw in that first study. But she also takes that pollen and nectar and feeds it to her offspring, and then her larvae will consume that pollen, and then they grow up and start that cycle all over again. So, if that pesticide exposure has impacts on later life stages, this is what we consider to be an ecological carryover effect, which is where an individual's past environment or experience can actually impact their current performance. This concept is really well documented in many organisms across a range of environmental stressors, especially for stressors like food or temperature. But it hasn't really been studied in pesticides in this way. So I was really interested in the potential for carryover effects of pesticides because pesticides are almost always studied at just one point in time at the time of exposure, despite the fact that that exposure could actually affect the performance of individuals and populations long after direct exposure, which could magnify those consequences. So to look at this, I did another cage study. So if you remember the cage study I described before, the flowers either had no pesticide on them or they were treated with pesticide. So you can see here in this illustration, the yellow flower at the top indicates a normal flower with no pesticide exposure. And then the green flower at the bottom indicates a flower that was treated with pesticides. And if you remember, these bees foraged on these flowers and they laid their eggs in nests. So mothers that were in these untreated cages fed their offspring pesticide-free pollen, and mothers in the treated cages fed their offspring pollen with pesticides. So we still have these two treatments, basically pesticides versus no pesticides. And then these bees overwintered in their nests as normal, completing the rest of their life cycle there. And then in the following year, those offspring emerged as adults. So what I did that year is I set up the field cages again, just like the first year, and I treated those cages again either with or without pesticide. So this provided basically every combination of pesticide exposure or no exposure over 2 years. So we can see what happens if bees are exposed multiple times, or if they were exposed in the past but not in the present, and how that actually impacts them in the second generation. So the second generation of bees again reproduced in these cages, and we measured their reproduction. And let's take a look at some results. So for the bees that were never exposed to pesticide, either in the previous year or in the second year, I'm going to represent those bees' baseline reproduction as just 6 offspring in the little icon for simplicity. So for bees that were only exposed in the second year, they had a reduction of about 30%. So they produced about 30% fewer offspring if they were only exposed in the second year. And for bees that were only exposed in the past year but not in the second year, they also produced about 30% fewer offspring. However, for bees that were exposed to pesticide at both times, so both as larvae and again as adults, their reproduction was even more reduced. So they had a 45% reduction in reproductive output from that double exposure. So this is really important to think about because this actually represents a pretty common exposure scenario for bees, especially for bees that live in agricultural areas. Pesticides are often applied multiple times throughout the year and year after year, so this repeated exposure is quite likely. And just like in the first study, we did some further research to get a sense of how this decline in reproduction could actually affect these populations over time. And so we created a simple population growth model to predict the effects of this pesticide exposure on population growth using these 2 years of data that we collected. And so for this model, we incorporated the overall reproduction as well as the offspring sex ratio. And we found that repeated exposure across 2 years, so that double exposure, caused an additional negative effect, especially when you consider the impacts on sex ratio, which reduced the population growth rate by 72% compared to unexposed populations. And this is a really striking, huge decline, and really indicates that carryover effects should be considered in risk assessment and conservation management, because we often see impacts that you couldn't see by just looking at one life stage or one level of exposure alone. And so, in summary, from this study, we found that pesticides can have really long-lasting effects, and a single pesticide application can reduce reproduction for bees the next year. So these studies that I just described were conducted in that field cage setting. I was also really interested in seeing if these trends were consistent in more open agricultural landscapes. So I did some follow-up studies in agricultural landscapes, looking at both solitary bee and social bee reproduction, to look at the impacts of both food resources and pesticide exposure in the field. And I don't have time to go into all this research today, but we did find similar results from the cages when we looked at these dynamics in the field. All right, so I just talked a lot about food and pesticide stressors. And another one of my big interests in research is the impact of climate change. And in this figure, I kind of have it as just one little piece in tandem with all the other types of environmental stressors that we think about in relation to bees. But really, climate change kind of exists alongside all of these and like kind of in and around everything as part of this really big environmental change. And so I'm really interested in studying the impacts of climate change on bees, specifically how warming temperatures affect bees and their interactions with the environment. So we are already seeing really distinct patterns of climate warming, both in terms of rising average temperatures, but also in terms of increases in the intensity and frequency and magnitude of these extreme heat events, heat waves. So these are all recent headlines, and in the past few years, we've continued to break heat records globally. And heat waves are particularly concerning because they have these really acute, intense impact on bees and all organisms living in the environment. In the Western United States, as just an example, heat waves have increased by about 300% in frequency over the last 25 years, and these trends are consistent in other areas as well. Here's some data from California showing the number of heat waves historically in the state, as well as some heat wave projections over the next 70 years. So we can see this follows a lot of other climate trends where we're going to see increased warming, increased extreme heat events. But we're especially concerned about the impacts of heat waves on bees because they are ectotherms and many species have relatively limited thermoregulation abilities, especially during immature stages when they are confined to their nests in those immature life stages. So I wanted to introduce an amazing undergraduate student who I worked with on this study named Gigi. After we experienced some really intense heat waves in California, we were both really interested in studying their impacts on bees. And we were kind of thinking about impacts throughout the whole life cycle, and we realized that there was hardly anything known about how temperature affects early larval development in bees. This was really surprising to us because we know that this larval life stage, sort of that first stage right after they hatch out of their egg, is a particularly vulnerable time in a bee's life. They are confined to a nest, so they can't behaviorally escape heat by going somewhere else. They're also really prone to desiccation. They can dry out really easily. And all of this is really concerning in the face of climate change because these early spring heat waves are becoming more common. So this could add even more stress to this already vulnerable life stage. So we designed a study where we exposed bee larvae to two different intensities of heat waves. We chose a mild heat wave and a severe heat wave compared to control conditions with no heat stress. And so I'm going to show you some of our results here. You can see on the y-axis, we're looking at larval mortality, so the proportion of bees that died from each of those treatments. And the temperature is shown here by color and shape. So for the control bees that did not experience a heat wave, they experienced pretty low mortality, just to be expected. There's always a little bit of mortality over the year, but it was pretty low for control bees. That mild heat wave at 31 degrees Celsius also had no impact, no difference from control conditions. But when we look at the severe heat wave, we saw a really dramatic increase in mortality, where over 75% of the larvae that went through that heat wave died from exposure. And so this really showed us that bee larvae can die from spring heat waves and in pretty large numbers, which is really quite concerning for the future of the species under climate change, especially if heat waves hit at that really sensitive larval time in their life cycle. And so we wondered if these heat waves are having this big of an impact on larvae, this very early life stage in bees, what happens when they get a little bit older? So we typically think of that next life stage, the pupal life stage, as being more resilient to temperature stress. But Osmia reach that pupal stage during the peak of summer, when heat waves are more intense and generally have higher absolute temperatures. So we were really curious what would happen to bees exposed to summer heat waves during that pupal stage of development, considering that those heat waves tend to be even hotter than the earlier spring heat waves. So we designed a very similar study where we exposed bees to three different treatments. So they had control conditions, a low intensity heat wave for 5 days, or a high intensity heat wave for 5 days that we started once they hit that pupal stage. And this figure here shows us the proportion of bees that survived overwintering. So they survived not only the heat wave, but they also survived through the rest of the summer and successfully emerged as adults the following spring. So we see that heat wave exposure also significantly reduced Osmia lignaria survival to adult emergence the following spring. So only 37% of the bees in that high intensity heat wave treatment in red survived to emergence, compared to about 80% in the low intensity heat wave and 91% in control bees. So we can see here already that this 5-day summer heat wave, even though it hits during a more robust stage in the bee's life cycle, can still have really dramatic impacts on bee survival. One potential reason for this decline is that bees exposed to heat waves burn through their fat body reserves. So the fat body is the primary location for energy storage in bees. It's really essential for sustaining their metabolism throughout development and throughout the winter. And so to measure the fat body condition in these bees, we x-rayed bees at the end of the summer while they were still in their cocoons. So you can see here on this X-ray image, these are each of the nests. And this is not an image from our study, but this is another X-ray of osmia nests. So you can basically see into all of the cocoons. And so these bees on the right, these are bees from our study, and we can basically see inside each bee and what their fat stores look like. So we scored each bee on a 3-point scale. So 0 indicates no fat body depletion. You can see on this image, they look pretty completely white with almost no fat body loss. We look right in the kind of this area in the abdomen. 1 is the score they received for moderate depletion. You can see in the abdomen, they're starting to lose some of that fat body. It looks a little darker here. And then 2 was the score for severe depletion. So you can see here most of this abdominal area has darkened, showing a pretty extensive loss of that fat body. And so this figure here, I'm going to show you the predicted probability of bees in each of these treatments falling into those 3 categories. So in the control group on the left, these bees did not experience any heat wave. Most of those bees had little to no fat body depletion. We do expect some fat body depletion over the summer, just because bees are using these energy stores during development and preparing for overwintering. So this is a pretty expected result for that control group. We saw a little bit more of a shift towards severe depletion in bees exposed to a low intensity heat wave. But in the high intensity heat wave on the right, the vast majority of bees, about 85% of bees exposed, showed a severe depletion. So heat waves likely increase metabolic demands on bees, which leads to a faster depletion of fat reserves and basically leaves fewer resources available for overwinter survival and reproduction. Because if you remember from their life cycle, these bees eat a lot, that whole provision during the larval stage, but that's all the food they get for the rest of development. So they really need those fat stores to power them through the rest of the year. So these sublethal energetic costs could amplify the effects of heat waves, as even bees that survive that initial heat wave will enter diapause, that winter diapause period, with compromised reserves that could impact their fitness or even their survival through the winter. And in addition to impacts on bee fat bodies, there are a lot of other normal physiological processes that are disrupted by extreme heat exposure. One of these disruptions is impaired protein stability. So this means that essential proteins in their bodies start to denature and stop working properly. And so a common way to study this heat stress response at a more molecular level is to look at the heat shock protein HSP70. So these proteins help to stabilize and repair other proteins that are damaged by extreme heat. And so we were really interested in looking at some molecular responses to heat stress. So we used qPCR to look at this HSP70 expression in these bees during this same heat wave experiment. So this figure here shows the expression of the heat shock protein HSP70 across different time points before, during, and after the heat wave. So if you look at this x-axis here, we measured their gene expression before the heat wave, on the first day, the second night, and the last day of the heat wave. And then we also took a look at it 5 days after the end of the heat wave. So here you can see the blue bars indicate the samples taken before and after the heat wave, and the red bars are the samples taken during the heat wave. And so in this low intensity heat wave that I'm showing you here, the expression of this heat shock protein increased pretty gradually. It peaked on day 5 on that last day of the heat wave, and then it did return to baseline after the heat wave ended. And if we look at the high intensity heat wave, the response was much sharper. So expression spiked really quickly, and it peaked kind of on early on in the heat wave, on the second night of the heat wave. It had already started to decline by the last day of the heat wave, even though the heat wave was still ongoing, and then it returned to baseline after the heat wave ended. So we can see here, these bees are clearly mounting a molecular stress response to the heat wave, but the kind of transient nature of this response suggests that there could be limits to how long this defense can be maintained. Um, and I think most importantly, even this strong molecular response in that high intensity heat wave wasn't enough to prevent that high mortality that we saw in those bees. So this was just a smaller part of a larger molecular experiment that we conducted where we were looking at RNA sequencing data from hundreds of different bees. We're currently analyzing this data, but I think this represents a good first look at what is happening to these bees on a molecular level throughout heat wave exposure. So when we think about the impacts of heat waves on solitary bees across their life cycle, we see that early spring heat waves have a pretty dramatic impact on larval survival, with over 75% mortality of bees in the high intensity heat wave. And we also see a pretty high mortality of summer bees, or summer heat waves, at that pupal life stage, with over 60% of those bees not surviving to adult emergence. And this is really important because the heat wave temperatures that we used in these studies are field realistic, meaning that they are already happening in the environment and they're projected to increase in frequency and intensity over the next 50 to 100 years. So these studies show us that heat waves could be a major driver of bee decline or bee stress going forward, and that we should definitely continue studying their impacts so we can better understand what might happen and how we can better protect bees in this climate future. So this work could not have been done without a huge community of people. I want to thank everyone who helped with this research, especially all the undergraduate research assistants and independent researchers who have worked alongside me, my co-authors and collaborators, and my funding sources. And I'd be happy to take any questions.
Thank you, Clara. That was fantastic. I see a couple of questions already in the Q&A box. So the first one is by Chris. He asks, it looked like Osmia lignaria only produced 40% female offspring under ideal or high resource conditions. Is that correct? Was he reading the chart correctly for that?
Yes, yeah. Osmia lignaria have a relatively low reproductive rate compared to other insects, but it's I think probably middle of the road for solitary bees.
Okay. The next question I see by Blair. She says, apologies if you explained this, but how did you heat the bees for your second experiment? Was it done in the field or was it done in the lab? And how did you heat them? Actually
Great
heat them?
question. Yeah, great question. So for both of those heat wave experiments, we had bees flying out in the field. So we had the adult bees nesting, and then we brought the nests into the lab, and we put the nests into different temperatures of incubators in the lab for the heat treatment. Yeah, so it was like the nests were produced out in the field, and then we brought them in for the heat treatment.
Oh. The next question by Molly. For your pesticide research, what pesticides did you test and were they insecticides or herbicides?
Great question. I used the systemic neonicotinoid insecticide imidacloprid. I used a soil drench of the insecticide to the soil um, during flower growth at sort of basic label rates.
Okay, another question by Tova. She's wondering if you know which life stages the offspring were likely to die at for pesticides and heat exposure. Is it eggs, larvae, or pupae?
That's a great question. For the pesticide studies, I did not look at the bees during basically during any of the development or overwintering stages. I just measured whether or not they emerged in the spring. So I don't know for the bees that died at what life stage that happened. I do know that a big difference was just that there were fewer bees even like there were few offspring even laid in nests. So the big impact there was just lower offspring even produced. There wasn't as much mortality. For the heat wave studies, the first study we basically were monitoring the bees um, every single day to see when those larvae were dying, and the majority of the mortality happened within the first couple of days of heat wave exposure. So it was a pretty quick, if they were going to die, they were most likely to die right away. And then if they were able to make it through the first few days, then they had a higher chance of survival. With the pupae, we weren't able to, because they were inside their cocoons, it's harder to tell exactly when they die through an X-ray. So we only measured them at the end of the summer and then at the end of
Mhm.
overwintering.
Okay. Molly has another question. If the repeated pesticide exposure causes such dramatic and rapid declines, wouldn't this lead to local extirpation quickly? How are any bees persisting in agricultural landscapes then? Are they recolonizing from adjacent habitats?
That's a great
Yeah.
question. There have been documentations of kind of really massive bee die-offs from some pesticide exposures and applications that have been actually connected to those applications. I think the scenario that I provided in my study was sort of a worst case but field realistic scenario. So the bees were in cages where they didn't have a choice on whether they had pesticide exposed flowers or non-pesticide exposed flowers. Generally in the field, there's kind of a more of a mixture of different types of active ingredients and different levels of exposure depending on what bees are foraging on. And so they're kind of integrating risk from a larger area, larger different types of things. And so that's one of the things that we saw in the field study was that there were impacts of these pesticide mixtures and the different levels that did still impact bees. But I think, yeah, I guess the answer is it does happen, but it's less likely the exact scenario that I provided because there's a little bit more sort of mixing of other types of things in the environment.
Great. And I guess touching on that, I'm just going to sort of jump into that question. Because they're forced to either forage on untreated plants or treated plants, did you notice that there were actual foraging differences, like the time they would forage and stuff for the ones that were on the pesticide flowers, or do they just forage equally when they're given no choice?
Yeah, that's a great question. So I didn't actually do any measurements of handling time or foraging, things like that. I did measure foraging trip like the number of foraging trips and the length of the foraging trips. And there was impact. I believe that pesticide exposed bees took fewer foraging trips overall, but those trips were longer. So that
Hmm.
suggests a reduced efficiency in foraging, or just a reduced motivation to go out and forage. And so I think, and there are other studies looking at impacts of exposure on foraging, and there tend to be impacts a little bit on cognition and learning depending on the type of
Hmm.
exposure. I didn't measure things like handling time or learning in this study.
Okay. Next question by Blair. If there's time, could you please touch on your field experiment that you didn't have time to cover today?
Sure, yes. Okay, so I have essentially we're trying to mimic the same sorts of stressors, but in a broader field environment. So we did the study in an almond orchard system where we had 10 almond orchards. Half of the orchards had a really big wildflower planting. So I think I have a slide here. You can see this is a really big wildflower planting where there was a big wildflower strip adjacent to the almond orchard that provided floral resources that were not directly treated with a pesticide. And then we had a range of different types of management of those orchards to kind of provide a range of exposures. And we measured both the impacts of those floral resources on both Osmia lignaria and Bombus vosnesenskyii, the common bumblebee in California where we were working. Um, and so basically the impact of these floral resources was that they increased food resources and decreased pesticide exposure by providing a pesticide-free forage. We did find that the floral plantings were extremely important, especially for the solitary bee reproduction. There was almost no reproduction for solitary bees at the orchards that did not have a almond orchard. For bumblebees, it was a little bit different because they have a longer foraging range. They were able to integrate resources from a larger area, but they still had a really big impact of the pesticide load. So the higher pesticide exposure they received, which we measured from the pollen within their nests, the lower reproduction that they had.
Great. Okay, another question by Christian. Could you talk a bit more on the time-dependent pesticide exposure effects on reproduction across life stages? So exposure at first year only versus second year only had similar reproductive effects.
Yes. Okay. So let me go back to that figure. There's a lot going on in that because there's so many combinations of exposure.
Yeah.
So did you have a specific question on which, like, any specific combination, or just wanted to know more in general?
He did state that exposure, well, I think he was looking at first year only versus second year looks like it had similar reproductive effects.
Yes.
Christian, if you want to
Yes.
expand on that, feel free.
Yeah, so this was actually quite interesting that the basically a single exposure, whether it was to the foraging adults or whether it was to larvae, had a really similar impact on reproduction. So if bees were exposed as adults, it had a really similar impact to when they were exposed as larvae. There's a lot of possibilities for this. We know that the larvae are eating more of that contaminated food, but they also might have a longer time to like I guess what we also only use the survivors from that exposure. So if larvae were able to survive through the rest of their lives from that exposure, then that they had a relatively similar impact to adults that were just kind of exposed acutely right before reproduction.
Okay, because he did also ask, so weren't there any life stage dependent effects? So
Life stage dependent effects. Do you mean like impacts on larvae that were different from impacts on adults?
I believe that's what he's asking. Yeah.
Okay, yeah. So we were looking at the only output we looked at was basically reproduction, the whether they initiated nesting, how many offspring they produced, and the sex ratio of those offspring. And the biggest impact we saw was from the adults that received the exposure, and the second biggest, yeah, was from the larvae that received exposure. We didn't look at anything that was happening over the winter, so if there were developmental impacts on the bees. We wouldn't be able to detect that because we only looked at whether they survived to emergence.
Mhm. Okay. We have one more question. So how did you individually mark the female bees in the field cages, and was that with paint?
Great question. Yes, I used Tester's enamel paint. I think I have a photo here. It's paint that's used for painting, I think, like little miniature railroad cars, trains. But it works really well because it's weather resistant. So it doesn't hurt the bees and it also stays on there through all the rain and weather and things that they experience. I used different combinations of colors so that I could tell the difference between them. There's all people sometimes use like Posca paint pens. I think those work pretty well. They're a little less durable outside, but there's a lot of different ways that people have found to mark bees. I thought this was the easiest for me. Yeah, and I painted them in a cold room, so um, they didn't fly away while I was trying to paint them, and I think it reduces stress. So just kind of chilled them down for a minute, put the paint mark in, and then release them outside.
That's great. Okay, I don't see any more questions. We have a little bit of time, so if anyone wants to ask one more question, feel free to ask it now, but I will just start sharing just a couple ending slides here. Thank you so much, Clara, for your talk. It was fantastic.
Yeah, of course. Thanks for all of your questions.
Okay, I don't see any questions. Oh, okay. So just before everyone does leave, thank you again for attending. I only just have a couple more slides to share. And so I just want to set another reminder that we do have registration open for our next Bee speaker, which will be in November, so not October. It's by Dr. Alexander Walton. You can register for his talk now. That link is ready. Also, just in case, I would like to remind everyone that we do have our BeeCon event coming up in a month in October. Our keynote speaker is Dr. Jessica Forrest. She is a full professor in the biology department at the University of Ottawa, and she'll be giving her keynote talk on the Friday of BeeCon. So that's the in-person day at York University. And just for those who may not know, BeeCon is a 2-day event where the first day, Thursday, is a virtual day, so all talks are presented through Zoom platform. And then Friday, we have all in-person talks. But those talks will also be streamed live for viewers who want to attend virtually. And so we also do have about 40 additional talks from speakers from different countries that will be presenting. Again, registration is still open. It'll end on September 30th. And oh, there's one more question. Let me pull that up. Okay, one last question by Henry. So have you considered to do this kind of pesticide wild bees relation in neotropical regions? And do you consider the responses or negative effects will be stronger in this region?
That's a great question. Yeah, one major limitation of this study is that I just studied one bee species in one context. I think this definitely should be studied in neotropical regions. I don't know what the major pesticide risks are in those areas, but I think it would be great to try and match the types of exposures that are really common to the types of bees that are there. I think that would be great. I don't know. There's so many differences in the species and the active ingredients and the climate that I don't know how that would differentially affect bees, but I would love for that research to be done.
Okay, I don't see any more questions. So thank you everyone for joining us today for our first talk in the BEEc Speaker Series, and we'll look forward to seeing you again in November. Thank you, everyone.
Thanks, everyone.